Cell signaling. Cell signaling typically communicates using cell signals, and these chemical signals, which are proteins and other molecules, are produced by sending cell, which is often secreted from the cells of our body. These signals float as messages through our body as signals, and it goes on passing to the nearby cells. Whereas the sending of this signal takes place by a ligand and receptor. The sending cell gives out ligand, whereas the target cell has a receptors, where this ligand goes on binding the receptors, and a non-target cell, the non-target cell do not has receptor, and this ligand doesn't go and bind to the non-target cell. The response comes only from the cell which are targets and which has the ligand attached to the receptors. Next, in cell signaling, the messages are carried by ligand are often relayed through the chain of chemical messengers, and this ultimately can be between intracellular and extracellular, and intracellular is within the cell and extracellular is between the cells. And cell signaling takes place and ligand attached to the receptors, whereas other cells organelles receive the signal from the receptors, and the response comes out from the cell. The forms of cell signaling can be divided about the neighboring cell or the target cell, whereas if the cells are near, it can be divided into a separate name called a paracrine signaling, and whereas if the target cells are long enough, they are called as endocrine signaling. And the forms of signaling are of four basic categories, and which includes paracrine signaling, autocrine signaling, endocrine signaling, and signaling by direct contact. In the direct contact of cells, they can be between the gap junctions, and in plants it can be of plasmodesmata. And the main difference between the categories of cell signaling is the distance where the signal travels. In paracrine signaling, the cells that are near often communicate through the release of chemical messengers, and these cells communicates each other with a relatively short distance is called as paracrine signaling. And in paracrine signaling allows the cell which are locally coordinates itself. And the example for paracrine signaling is a friend from a first bench which who are talking to a second bench guy. They are neighbor to each other, and the signal passes only between them, or the communication takes place only between the first bench and second bench members. When the member of one group of cell takes to the neighboring group of cell is an identity which should be taken on. Synaptic signal. Synapses takes place mainly in nerve cells, whereas nerve cell releases signals and which the axon of the other nerve cell receives the signal, and this is called a synaptic signaling. The signal sending in the neuron fires an electrical impulse which travels on the fiber extension called as axon, and this impulse reaches the synapses and then triggers the release of the signal through neurotransmitters. When the neurotransmitters arrive in the receiving cell, they bind to the receptors and causes a chemical change. And this is the overview of it. Whereas the neurotransmitters release the signals, which on takes place by a neurotransmitter here. The neurotransmitters that are released in the chemical synapse quickly are degraded or taken back into the sending cell. This is called as resets of the system to the next cell. In paracrine signaling, the cell targets the nearby cell, whereas in autocrine signaling, the cell targets its own. The targeting nearby cell takes place to the signal which is transferred from one cell to the other cell, whereas in autocrine signaling, the cells its own targets itself. And in autocrine signaling, the ligand which has been produced by the cell gets targeted into itself. It is mainly it can be takes place in cancer cells for the process of metastasis, and the signal which has been transferred to itself gets on bound to the receptor of its own cell, and it gives out the signal. And in endocrine signaling, the cells transmit signals over a long distance through the bloodstream, whereas it can go and bind to the target cell's receptor, which is far away, and it also takes place in the process of hormones. In hormones, they include thyroid, hypothalamus, and pituitary, whereas gonads and pancreas. Each endocrine gland releases one or more type of hormones, which can be later into the process of development. In endocrine signaling, the signaling cell target goes on to the bloodstream, and the cell targets a distant cell to the bloodstream. The example for endocrine signaling can be given as friends in the opposite sides or in the opposite directions of the class. The signals can be transferred via a rocket, or else via messages, or via call, whereas in our body it takes place via the bloodstream. Prokaryotic cell and eukaryotic cell. The difference between prokaryotic cell and eukaryotic cell. Prokaryotes means in a Greek word it is termed as pro, and it can be known as before nuclei. The prokaryotic cells are very much small when they are compared to the eukaryotic cell. Prokaryotic cells, the example for prokaryotic cells are bacteria, archaea, those kind of cells, and this is the outline of prokaryotic cell. It consists of a bacterial cell anatomy where it has pillars, capsule, cell wall, plasma membrane, cytoplasm, nucleus, and ribosomes, plasmid. Flagella and pili are used for the process of locomotion, whereas the nucleoid, it does not contain a well-defined nucleus, so it can be divided in the main division of classification between a prokaryotic cell and the eukaryotic cell is the um, existence of the nucleus or a nucleolus. In a prokaryotic cell, the other defining characteristic of prokaryotic cell is that it does not possess membrane-bound organelles such as nucleus. Reproduction takes place as binary division. Binary division has an example of amoeba in which a parent cell divides into two daughter nucleus. Structurally, prokaryotic cell has a capsule enveloping a body which functions as a protective coat, preventing the process of phagocytosis. Phagocytosis is the engulfing of microbes, and the capsule helps the cell to escape the process of phagocytosis. Pillars and flagella are the locomotory organs in various environments, and pillars is essentially resists being fleshed, and hence it is also called as attachment pillar. It is commonly observed in bacteria. Right below the protective coating lies the cell wall, which provides strength and rigidity to the cell. Further down, it has a cytoplasm, and it helps the cellular growth, which contains a plasma membrane. The plasma, it separates, the cytoplasm separates the interior contents of the cell from the exterior environment. When within the cytoplasm, ribosome exists and it plays an important role of protein synthesis. It is also one of the smallest component of the cell. Some prokaryotic cell contains a special features called as mesosome. Mesosome is an organelle which is involved in the process of cellular respiration. Most prokaryotes contains the plasmids and smaller circular species of DNA, which is smaller circular species of DNA which can be undergone for the process of recombinant DNA technology. To help the locomotion, flagella is present to the pillars, and it also serves as the aid of locomotion. Common example of prokaryotic cell, it comes under the kingdom of Monera and Archaea. In kingdom Monera, or in the five kingdom classification, we have Monera, Protista, Fungi, Plantae, and Animalia, and Monera, maximum members of Monera comes under prokaryotic cell division. Eukaryotes. Eukaryotes can be also called as a cell which has good or true nucleus. Eukaryotes are the more complex and they are much large cell. They contain all major components of the cellular system. And animal cell. In an animal cell, it contains various different organelles such as peroxisome, centrosome, centriolysosome, ribosomes, and much more. Whereas it contains cilia, cell membranes, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, etc. Structurally, eukaryotes possess cell walls which supports the plasma membrane. Cell walls gives supportment to the plasma membrane, whereas the cell is surrounded by plasma membrane. It controls the entry of certain substances in and out of the cell. Nucleus contains DNA which is responsible for strong genetic information. DNA has genetic information which has been passed from our ancestors till date, and the nucleus is surrounded by a nuclear membrane. Inside the nucleus, we have nucleolus which plays the role of synthesis of proteins. Eukaryotic cell, it also contains mitochondria, chloroplast, and endoplasmic reticulum, etc. And mitochondria is also called as a powerhouse of the cell, which generates ATP. Chloroplasts are seen only in plant cells. Chloroplast gives a chlorophyll pigment, which is used for the process of photosynthesis. Endoplasmic reticulum gives or helps in the process of transportation of certain materials, whereas other cell organelles such as ribosomes, Golgi apparatus, vacuoles has their own process. Centrosomes are used for the process of mitosis or meiosis, whereas vacuoles are used as a storage hub of the cell. Cytoplasm holds cells at the position, and example of eukaryotes are most unicellular organism within the nucleus and also the multicellular organism. The characteristics of unicellular and multicellular organism can be divided, and the main process is nucleus is present in eukaryotic cell, whereas it is absent in prokaryotic cell, and the growth of growth rate is slower in eukaryotic cell, and in prokaryotic cell it is faster. Organelles are present in eukaryotes and they are absent in prokaryotes, whereas chromosome they are more than one in eukaryotes, whereas the prokaryote it is a single long loop of DNA. It is coiled into one another. These are the differences between prokaryotes and eukaryotes, and in the main difference between prokaryotes and eukaryotes is as prokaryote does not have well defined nucleus, and in eukaryotes it has a well defined nucleus. Into the next topic, it is cell cycle and cell growth control. Cell cycle and cell growth control. To divide a cell must have a complete several and important task. It must grow, it must copy its chromosome, and it should be divided. And the cell must have it should get divided into two daughter nucleus. And a series of steps makes this process of dividing of a cell into two daughter nucleus. It's a process of called a cell cycle. And the cell cycle is a cycle rather than a linear pathway. It is a go around process. It does not get stopped. In eukaryotic cell, it has two major phases: interphase and mitotic phase. During interphase, the cell grows and it makes a copy of DNA, it makes a copy of organelles, and in mitotic phase, it gets divided. The cell itself gets divided into two. Interphase. G1 phase, S phase, and G2 phase. G1 phase is known as gap phase 1, G2 phase is known as gap phase 2, S phase is known as synthesis phase. In G1 phase, the cellular contents excluding the chromosomes are duplicated, whereas in the synthesis phase, all the 46 chromosomes are gets duplicated. In G2 phase, the cell checks itself whether I can go for a mitotic phase or a meiotic phase, whereas in mitosis and meiosis, the cell gets divided into two phases. During G phase 1, it is also called as first gap phase. The cell physically grows itself, copies its organelles, and makes a molecular as a binding blocks. In S phase, synthesis of chromosomes takes place into 46. And next, in G2 phase, the cell grows more and makes the proteins and organelles. It begin to recognize the contents and prepare it for mitosis. At the end of G2 phase, mitosis phase takes place. Mitosis is the attachment of a mitotic apparatus, and it separates the sister chromatids into two of the partitioning of genetic material. The separation of the genetic material in the nuclear division is the nuclear separation is called as karyokinesis, and the cytoplasm separation is called as cytokinesis. The phases of mitosis are prophase, metaphase, anaphase, and telophase. In prophase, the spindle fibers appears and the chromosomes condense, and in metaphase, the chromosomes gets aligned at the equator, and in anaphase, the chromosomes get separate into two, and in telophase, the nuclear membrane reforms, and the condensed chromosomes decondense, and in telophase, the cytoplasm divides into two genetic material. In prophase, it occupies half the mitosis. The nuclear membranes break into small vesicles, and the structure known as centrosome duplicates to form two their daughter centrosomes. The centrosome organizes the production of microtubules, and which in turn releases mitotic spindle. This mitotic spindle gets attached to the condensed sister chromatins, which is held together with structure known as centrosome and centromere. The chromosome is led by the centromere, and it migrates to the equatorial line at the prometaphase. The region of mitotic spindle is known as metaphase plate. And in prometaphase, the spindle fibers gets bound to the associated with the centromere of each chromosome called as kinetochore. The individual spindle fibers bind to the kinetochore structure. The chromosomes continue to condense. At the metaphase, the chromosome align themselves at the metaphase plate of the spindle fiber apparatus, and at anaphase, the sister chromatins, which are four arms, gets divided into two arms, and it goes to the opposite poles at the end of the region. The separated sister chromatids are now referred to as daughter chromosome, and the nucleus gets divided. In telophase, the final stage of mitosis, the reversal of many processes observed during the prophase. The nuclear membrane gets reformed, and they get divided into two nucleus in a single cytoplasm. Whereas in cytokinesis, the cytoplasm gets divided into two so that they form two different cells. In mitosis, in prophase, the sister chromatids gets condensed. The centromere takes place into two places. It is a metaphase plate, and the centromere gets attached to the kinetochore, and they get divided. During anaphase, the sister chromatids get separated, and at telophase, they are divided into two. Meiosis. Meiosis takes place separate part of homologous chromosomes. It mainly takes place in homologous chromosome. The special cell division it forms diploid to haploid. The homologous chromosomes pair up into exchange of DNA into recombinant proteins, and there are five stages in prophase. They are leptotin, zygotin, tachytin, diplotin, and diakinesis. In leptotin, chromosome starts to condense. In zygotin, the homologous chromosome, there are closely related chromosomes, get attached to each other to form four different chromatids. In packeting, the crossing over between the homologous chromosomes takes place. The overlapping of the chromosome takes place, and the process where the overlapping takes place is called crossing over place or chiasma. In diplotin, the homologous chromosome starts to separate but remain attached at the place of chiasma. In diakinesis, the chiasma gets separated and they become different chromosomes. In prometaphysis, spindle fibers, the same process in mitosis takes place. The spindle fibers get attached to the kinetochore. In metaphase, they get aligned at the equatorial line, and in anaphase they get separated into two to the different poles. In telophase the chromosome becomes diffuse and the nuclear membrane gets reformed. In cytokinesis the final cellular division into two new cells forms takes place. It is followed by meiosis 2. It is a process of mitosis. Prophase 1, whereas the chromosomes get condensed. In metaphase 1 they get aligned in the mitotic plate, and in anaphase 1 they get separated, and in the telophase they are formed into two nucleus. In the meiosis 2 process, prophase 2, the two different cells are then again aligned at the equatorial line. They are then changed into two different parts of the equatorial line, and they are divided into two. Whereas the in meiosis we get four daughter cell, which is single cell. Cell growth and cell growth control. If a cell has an error in its DNA, and that cannot be repaired, and error in the DNA cannot be repaired, and it should undergo self destruction. Self destruction is also called as programmed cell death or apoptosis. Apoptosis is a common process throughout the life which helps the body to get rid of the cell which is no longer needed, and it undergoes apoptosis by a cell called as macrophage. And cell growth control. If we have a damaged cell with a damaged DNA, and if the cell undergoes division of cell cycle, and when the number of cell increases, it causes tumor cells, and it takes on the process of cancerous reagent inside our body. Apoptosis protects the body by removing the genetically damaged cell, which could lead to cancer and the development of embryo and adult tissues. The disruption of a normal regulation of cell cycle leads to diseases such as cancer. When the cell cycle proceeds without control, when a cell with a damaged DNA carry on without the control, the genetic errors takes place in the cancerous tumor. And that is the end of cell cycle and cell growth control. Next takes place on cell death. Cell death and modes and mechanisms. Cell death. Cell death is an event of the biological cell ceasing to carry its function. I mean, the cell which cannot carry out its function should undergo cell death. It can be of two forms. It can be whether of apoptosis or necrosis. Apoptosis is known as suicide, and necrosis is known as killing of cell. Apoptosis has another name of programmed cell death, and necrosis is a process of decay and destruction. Apoptosis. It's a form of cell death in which the suicide program is activated within the cell. The process takes place on fragmentation of DNA, the cytoplasm will get shrinked, and the membrane changes, and the cell death without lysis or damage or to the adjacent cell. If a damaged cell is surrounded by a series of different cells, only the damaged cells undergoes apoptosis. The mechanism of apoptosis takes place into two phases: initiation phase and execution phase. In initiation phase, it takes place when the apoptosis enzymes are getting activated, and in execution phase, activated enzyme causes the cell death. Just make it clear: in the initiation phase, it takes place on the activation of the enzymes, and in execution phase, the activated enzyme causes the cell death. There are two types of pathway in initiation phase. It is extrinsic pathway and intrinsic pathway. In extrinsic pathway, different types of enzymes such as procaspase 8, caspase 8, caspase 3 goes on this process. And TNF is a receptor, whereas the death domain, which is in orange color, and a TRAD and blue color, these all gets activated when a ligand gets bind to the TNF receptor, and this FAD get activates the procaspase 8 enzyme, and procaspase 8 gets changed into caspase 8, and then caspase 3 gets activated, procaspase 3 gets activated, caspase 3, and it goes on the process of apoptosis. Inside a plasma membrane, these all process takes place during a cell death. Next, in extrinsic pathway, it happens in another way, whereas if FAS receptor is found and FAS ligand binds to it, it activates FAD and a flip protein takes place, which in turn activates procaspase 8, procaspase 3, caspase 3 gets activated, and it takes place the process of apoptosis. Suppose an extrinsic pathway can in turn takes place into an intrinsic pathway. Intrinsic pathway is mainly for the process of mitochondrial regeneration or mitochondrial destruction. It takes place with the protein called as split protein is mitochondrial dysfunction. And in intrinsic pathway, the severe DNA damage attacks the activation of apoptic factors such as BAX. This BAX creates pores on the mitochondria, whereas cytochrome C gets released in the mitochondria from the mitochondria. This cytochrome C enters into the process of apoptosomes, whereas in this apoptosomes activates procaspase 9, which in turn activates procaspase 3 and caspase 3. In intrinsic pathway, we have procaspase 9, whereas in extrinsic pathway, we have procaspase 8. This is mediated by execution phase, which in which activated enzyme takes place is mediated by caspase 3 and caspase 6, which is activated by caspase 8 and caspase 9. Which they are activated, they break the cellulose cytoskeleton protein and nuclear matrix protein, which in results used in the breaking of nucleus. Necrosis. Necrosis is a sum of total morphological changes which is followed by cell death in the living tissue. If a cell gets damaged, it can recover and it should recover. If at all the cell does not recover from its damage, means it should undergo the process of necrosis if it has a progressive injury. The breakdown of plasma membrane, the breakdown of organelles, nucleus, and leakage of contents takes place in this process. The mechanism of necrosis takes place by the loss of metabolic function. It loses its integrity, cessation of the ATP production does not take place. The organelles swell and they become dysfunctional. The different schematic representation of apoptosis and necrosis is that the formation of bulbs takes place, the nuclear begins to break apart, and the small pieces and the organs also located in bulbs. The cell breaks down the apoptosis bodies and organelles still function in apoptosis. Whereas in necrosis, the cell membrane ruptures and releases cellular contents and organelles are not functional. And this is the end. Thank you.